My name is Shannon Cornett, and on behalf of the Brooker Imaging team, I'd like to welcome you to our Billerica, Massachusetts demo facility, and this live from the lab edition to demonstrate the timsTOF fleX MALDI 2 platform and how it is used for MALDI imaging and spatial omics. Before we begin, I'd like to just take a few moments to orient you to the timsTOF fleX MALDI 2 platform, just to showcase some of the key features that you'll be seeing shortly. You may have heard already about our platform for 4D omics, our timsTOF Pro and Pro 2 platforms that have become the new global standard for four-dimensional omics analysis. This system has a dual TIM cell, which delivers maximal sensitivity and 100% duty cycle. Collisional cross-section is the fourth dimension, and that adds to the confidence level for identifications and also the ability to do some other things, as you'll see. Passive enables the high-speed analysis, the more than 100 Hertz MSMS throughput rates, and all the benefits this brings to the proteomics, metabolomics, lipidomics world. And I bring all this up simply because the timsTOF Pro 2 is part of the timsTOF fleX platform. And so anything you hear about the capabilities of the timsTOF Pro, that is certainly possible with the timsTOF fleX. What the timsTOF fleX adds to the mix is the high-speed SmartBeam 3D MALDI imaging source, which delivers world-class zoom mode imaging down to 10 microns per pixel. As you see in the illustration there, the key element of the MALDI LCMS source is it's fully integrated, so there really is nothing to change over when you switch from one mode to the next. It essentially is instantaneous switchover. And all of this kind of combines with CCS-enabled imaging to create true pixel images where when you detect a signal, you know instantly and immediately where what cells originate the signals. I mentioned that the TIMS is useful for imaging, and that's certainly one of the powerful elements of the timsTOF fleX. The dual TIM cell carries over for imaging, as you see in the example here. Isobaric and isomeric species can be separated in the TIMS dimension, and then by virtue of being separated, then we can clearly differentiate them spatially as well. You can see the two highlighted bands distributing differently in the tissue example. Most recently, we have added a MALDI 2 option to the platform. And MALDI 2 is essentially a second post-ionization laser that serves to maximize the image sensitivity in MALDI mode. And you can see from the example there the dramatic effects that this MALDI 2 option can deliver. You can see the left half of the image collected in MALDI mode, the right half in MALDI 2 mode. And so clearly there is a benefit, not only in species that we would normally detect, but also MALDI 2 has been found to activate molecular channels that don't ionize quite readily by MALDI in the normal process. So not only does it enhance sensitivity, but it also extends the range of compounds that can be imaged. And so just to kind of wrap up, we've talked about the top four elements here, the components of the timsTOF fleX platform. This year, we're bringing to the table Skills Autopilot, which is a process for automating the measurement setup and creates an instrument validation and automatic calibration that really is designed to remove a lot of the guesswork out of the MALDI imaging workflow. And so it adds to the streamline and reduces a lot of the expertise burden and learning curve for improving or pulling MALDI imaging into your laboratory. Now, just one logistical thing here as we go along into the lab and you see some of the workflows in action. If you have any questions, please enter them into the chat box and be sure that you check off to send to staff. That way we'll be sure to see them. And then at the end of the presentation, as time allows, we will ask as many of these as possible and get you the answers you're looking for. So with that, I'd like to kind of introduce my colleagues who you see on the screen here, the Christina Silvescu and Kate Stumpo.
Hello, everyone.
Both application scientists in Billerica. Christina is going to kind of demonstrate as you see her sitting at the instrument, she's going to demonstrate the workflow from the acquisition standpoint. And then Kate will describe our software components that really take the advantage of taking all the information that the instrument produces and rendering that into useful information. And so with that, I would like to pass the ball over to Christina. So Christina, please take it away.
Thank you, Shannon. Hello, everyone. Welcome to the Billerica Demo Lab. We are here right in front of the timsTOF fleX instrument equipped with the MALDI 2 technology, as you can see. And we'll be talking today about the mass spectrometry imaging workflow, how we acquire a image data set, and later on you will see my colleague Kate Stumpo showing you how to process the imaging data set. As you've seen from Shannon's technical introduction, the timsTOF fleX, it's equipped with a MALDI 2 laser that enhances ionization efficiency, enhances sensitivity. It's also equipped with a trapped ion mobility cell that allows us to separate isomeric and isobaric species. These two features are very important in mass spectrometry imaging, especially when we are looking into biological samples, into the tissue environment, which is very complex at molecular level. We want to have these features to allow us to efficiently ionize the compounds and also to separate them. To perform a mass spectrometry image experiment, we first start with sample preparation, and you see here on the slide, you see here on the slide the entire imaging workflow. We'll start with tissue that we want to interrogate with mass spectrometry imaging. We cut thin sections through the tissue, and then we mount them on a glass slide. We use specific glass slides which are conductive, and you can see here in my hand, I'm pointing to the camera, our glass slides. I'm also gonna show them in the image later on, right? So we'll we'll mount the sections on the glass slides, and after that we will take an optical image of this glass slide with the samples. Next, we want to deposit an uniform matrix layer on top of the biological sample, and for that we use automated sprayer, and you see here the TM sprayer. And after we deposit the matrix on the tissue surface, we would load the sample in the instrument. So I'm going to take over now, and I'm going to share with you my screen so you can see. Okay, so as we were talking earlier, I hope you can see now the layout of our MALDI target, which with two glass slides here. It's the same one that I was holding up earlier. You see on the bottom we have the Intelli slides, which are specific to Bruker, these are already inscribed with the barcode to the right, you can see here, and it also have inscribed the fiducial marks, the teaching marks, which allow us to automatically co-register the IntelliSlide that you see here with our mass spectrometry image acquisition software. So in this way, we can identify the position of the sample on the targeted surface. On the top, you see another IntelliSlide with tissue samples, which was covered with an uniform layer of matrix. And this sample, this glass slide is ready for data acquisition. Now, both of them are mounted in our target carrier, and I'm gonna, okay, close this for now, and I'm gonna show you in the camera how we insert the target carrier inside the instrument, right? So it has only one direction. We inserted it into the MALDI door, and once it's in, the target is going to be docked inside the MALDI source, and we are ready for data acquisition. You can see here in the instrument, we have on the right side the MALDI source, and on the opposite side, we have the ESI source. You also can see behind the MALDI 2 technology panel here, the front panel, I have a syringe mounted on the instrument, which allows us to infuse into the ESI source a calibration mix. So you will see today how we use both the MALDI and the ESI source for tuning, calibrating the instrument, and for starting the imaging run. So once we introduce the target in the instrument, the instrument, the software, which is Teams Control. You see here, I'm sharing with you my Teams Control layout. This is the software that allows me to operate the instrument. It will automatically dock in the target, and during the docking, I can perform target profile. So I account for any Z dimension changes. It will automatically perform focus adjustment. So once all these things are done, you can see the target is in, the instrument is showing me the geometry of the target here with the position that are covered by the glass slides, by the two glass slides. On top, you have the MALDI camera, which shows me the tissue and the matrix deposited on top of the tissue. And obviously, the system is gonna allow me to adjust parameters for data acquisition. So after I inserted the MALDI target, I will maybe go into the tissue. I can move around on the tissue surface and maybe fire a few laser shots just to make sure that the instrument is working well, that I have adjusted the laser power intensity. I also can adjust at this point the mass range. You see here on the right side of the screen, a image of my spectrum that I generate every time I fire the laser. And on the left side of this panel here, it's the mobilogram. So I can adjust both the mass range or the mobility range from this setting over here, these settings over here on the left side of the screen. And then once I set up all these parameters, I can make sure that everything is ionizing well. I checked my histogram map here my heat map here on the upper right corner. So in the heat map, you see one spot associated with a pair of values, m over z and mobility. We are acquiring both these values simultaneously, and we can characterize the molecular composition based on these two parameters. Okay, so I set up my mass range and mobility range to acquire an experiment for small molecules and lipids. In this case, I adjusted my laser power. I think I'm getting a pretty good signal with my regular MALDI acquisition. So next, I would go to calibrate the instrument to make sure that I have a good calibration. You can perform that in the MALDI environment here with spotted calibrants, but today I want to show you how we calibrate in ESI, just so you see how easy it is to change in between the MALDI source and the ESI source. So to do that, I'm just going to go to the left side of the screen here, and I'm going to unclick the MALDI button, and you'll see that the MALDI panels are going to disappear, and we are already in ESI, in the ESI source. So under the source here, I'm going to activate the syringe pump, and then I'm going to monitor to see the tune mix infusing from the syringe pump over here. So we already switched from MALDI to ESI. We are already injecting the calibration mixed. I didn't have to even touch the instrument. We don't have to vent the source to change it. So it's just one click of a button. Now you can see that my tune mix and calibration standards are already appearing in my mass range over here on the bottom, and they also appear in the mobilogram. And I can go to the calibration button, calibration tab, and I will calibrate first the time of flight. So I have loaded here my tune mix calibration standards, and I just hit calibrate button, and once I get a good score, I hit accept, and the system is calibrated on the TOF mode. Next, I'm going to move to TIMS, which is the trapped ion mobility separation cell, and I'll do the same. This time I have the reference masses, but also I have the reference mobilities. So I hit calibrate again, and then once I get a good score, which I did, I accept the calibration, and now the system is fully calibrated in both the time of flight and the trapped ion mobility cell. And that's it. We can next go back to MALDI. So I can go to source and inactivate my syringe, and you'll see that the calibrant goes out, runs out, and then click on the MALDI button. And now the MALDI panels appear, and I'm already in MALDI, and I can go and fire the laser. And once I fire the laser, I should start getting a spectrum associated with that pixel. So that's it. We flip quite easily between the MALDI and the SI. I can show you again. So we are in MALDI. We have the camera. Unclick the MALDI button. We are in ESI. I activate the syringe pump. And then I already get the tune mix infusing through the ESI source, and I can calibrate and perform any other tuning or anything else I want to do here. And then once I'm done, inactivate the syringe pump and click on the MALDI button. We are in MALDI. You see here we have two other buttons. One of them is for TIMS, which is the trapped ion mobility. I had the TIMS on from the beginning. The other one that you see in the middle is the post-ionization. So when I activate this post-ionization button, I'm activating the second laser on the MALDI too. You will see that once the laser warms up, we'll be able to acquire a spectrum in MALDI 2. So I'm going to hit start. Okay, so now you can see here a spectrum in MALDI 2. So what we usually observe with the second laser, with post-ionization laser, we observe an increase in ionization, especially of multiple classes of small molecules. And you can see that the spectrum here in MALDI 2, it's much denser in the small molecule range, but we also see a lot of other compounds that we are that we have never been able to see before with regular MALDI. And I'm going to save this spectrum that I acquired with MALDI 2. I'm going to save it in my SUM buffer. So I'm going to just add it to my SUM buffer now. And then okay, and then I'm gonna go back to regular MALDI. So right now this is the spectrum for MALDI 2. I'm gonna go back to regular MALDI. So I inactivate the post-ionization, and then I'm gonna fire the laser to acquire this time data with our regular MALDI. So you can see already the spectrum quality disappears, especially in the low molecular weight range. So MALDI 2 brings a lot of information about small molecules. Now, if I look into details, if I go back to my MALDI 2, which I saved in my SUM buffer, so this is my regular MALDI, I'm going to switch to my MALDI 2 spectrum. You can see the difference in the small molecule range. When I zoom into the spectrum, and maybe I looked qualitatively at specific ions, for example, I know here at 810.59, I have a phosphatidylcholine, which usually ionizes with regular MALDI, but I also see at 810.68, 67, this is a glycosylated lipid, which usually does not ionize with regular MALDI. So we already see it in the MALDI 2. And if I go to my regular MALDI spectrum, you see that the glycosylated lipid, it's a glycosphingolipid, disappears from my regular MALDI. Right, so this is my regular MALDI. Go to the SUM buffer. This is my MALDI 2 spectrum. So we bring qualitative additions to the spectra and to the molecules that we are able to ionize from the tissue with our MALDI 2 post-ionization. Okay, so let's say you set up your parameters in Teams control, however you would like to acquire your data, and then you can save your method. And I have here a few methods that we share with our customers. And you can see we already have predefined tuning methods and calibrate and acquisition methods. You can acquire with our system data sets in lipidomics, in metabolomics. We have proteomics. Each of them have positive and negative ionization modes. With each of them, you can acquire with TIMS on, with TIMS off. You can acquire with MALDI 2 on, with MALDI 2 off. So it's very easy to access these methods. Then once you save your method and you are happy with your acquisition, we can move to fleX imaging. So fleX imaging is the software that allows us to set up image acquisitions So our fleX imaging software contains already few steps that are automated for the users. So what we would want to do is to load the optical image of the glass slide, right, that we took before we deposited the matrix. We'd load the optical image, the scanned image, in a dedicated folder, and then when we start the fleX imaging software, the wizard will automatically go and pick up that scanned image, co-register the optical image with the position on the glass slide in the instrument. So it's going to use these fiducial marks to automatically co-register and generate the co-registered optical image with the mass spec image. Now, after all these steps are done, we'll be able to assign regions of interest. But before I go there, I'm going to show you how the instrument has already assigned the fiducial mark. So I'm going to click on this position sample carrier, and let's say I want to make sure that the instrument is going to the correct position. I'm going to, let's say I want to go to this tip of the tissue up here. So I'm going to just click on there, and you can see the camera is gone. It's already moving to that position. So it's very easy, just clicking on the optical image to move into the tissue. This is a very important feature because if you want to be specific about the areas that you are trying to acquire, it's very easy to select it this way. Now, to assign acquisition regions, we are using this buttons over here, which allows us to add rectangular measurement regions or polygonal measurement regions. But I also have this magic wand, which actually, when I click on it, allows me to automatically find the edges of the tissue, so I don't have to sit here and manually delimitate the perimeters of the tissue. So it's very easy to define regions of acquisitions. And at this time, I'm not gonna acquire multiple regions. I'm gonna have only one setup, and I'm acquiring at 50 micron spatial resolution, and we are actually ready to go. So next, I'm going to just hit start, auto execute run. And when I do this, fleX imaging walks you through the wizard, and then we can go through the instrument and see it starting. We are going to show you next how we perform the data analysis with our skills and Metaboscape softwares.
Hello, everyone. This is Kate. Thank you, Christina, for the introduction. And let me get my screen up so I can show you my screen. All right. So I'm excited to show you how easy it is to use our software tools for MALDI Imaging. Today, I'm going to show you how to turn your data into meaningful spatial images, how to use Skills Lab, which is what's up on the screen right now, for untargeted discovery, and finally, how to use our newest software solution, Metaboscape, to annotate peaks in your various omics-based experiments. So as I'm starting with Skills Lab here, I'd like to give you a brief orientation as to what you're seeing on the screen here. So I have the tissue section optical image as the most prominent point here. And then I have the average mass spectrum down here on the bottom. The right-hand side has all of the various functionalities. I've got several tabs expanded, as I find it easier to leave open what I'm going to be using most often. The tissue section that was imaged is outlined in blue here, but one of the features in skills is that I can add or define additional regions. So as I click on this, you can see the pink region of the renal cortex is now outlined, and the green region of the renal pelvis is now outlined. I may have regions of interest based simply on the anatomical slice, or perhaps I have additional information that I can upload, such as H&E stains, IHC, or fluorescence information. And then I can toggle the view in order to define my new regions of interest. You may have also noticed when I did those clicks to those new regions that the spectrum down here changed colors as well. So those correspond to my active regions. Since I have blue, green, and pink currently active, then there are blue, green, and pink spectra representing each region. For a last bit of orienting you, I'm going to take you through some of the tools that are available to help with mostly untargeted analyses. So I'm going to start by talking you through how you can use Skills Lab to peak pick for you or find peaks. I'm also going to talk you through segmentation, component analysis, and finding discriminative values. So first, I've used the Find Peaks function from this menu to look at the most prominent features that are present. And I've already pulled up that peak list, and I have it named down here in the bottom right. I've generated all of the images. There were 87 images from my peak picking list. It took about 3 seconds to generate all of these images. I can click on a given mass to charge ratio and pull up the image in order to view the spatial distribution. When I do a targeted experiment, I'm going to go into the mass spectrum and click on my peaks of interest and make a custom peak list. But since what I'm showing you here is a discovery viewpoint, I use Skills Lab to help me pick my peaks. In addition to this feature, it also has something that we call automatic spatial segmentation, which is a hierarchical clustering method, which helps to group pixels that have similar molecular profiles. This is a useful data mining tool for quickly segmenting your image pixels into clusters that have similar molecular compositions, and presumably similar cell type. This is especially useful when you don't have prior knowledge of the tissue pathology. So after the calculation is done, which I did ahead of time, and it takes generally under a minute to do, I've selected my segmentation analysis, and you can see that the image on my tissue section has now changed. I notice right away that I have 2 different colored regions. So again, the segmentation is grouping together pixels with similar molecular profiles. At this first round of separation or segmentation, I can see that the red area is likely corresponding off tissue. So I've outlined slightly beyond my tissue margin. So I didn't use the magic wand tool for this example. And you can see that the green is on tissue and the red is off tissue. Something that I really like is I can click on that green region and I can create a new region of interest that's specifically on tissue. And I can save that, and then if I so chose, that's going to show up here in my region of interest, and I could work solely with that on tissue region for future calculations. For the moment, I'm going to go back to segmentation, and I'd like to show you what else can happen here. So as I go further into the segmentation, I tend to call this the segmentation tree. I can start to view additional areas that have similar molecular fingerprints. You can see that in this kidney tissue section slice, I again am grouping based on the tissue architecture. So the molecular profile for each of the pixels are actually similar and showing up within this segmentation analysis. Okay, so I've shown you several tools here, but now I really need to start asking myself a question. How are things similar in these regions? What are the differences or what is changing? So I'm going to unclick the segmentation and I'm going to go back to this peak of interest that I highlighted, mass to charge 634. And I'm going to look at how I can compare my two different tissue regions, so the green and the pink. So first, I'm going to look at the intensity box plot. This is showing up for the full region, the blue, and then those two additional regions that I highlighted. First, I have a box and whiskers plot that shows up for each region. This represents the median peak intensity. That's the line across the center of the box. And then the second and third quartiles are the top and bottom. And then the whiskers are representing the upper and lower quartiles. The other plot that you see here is what I tend to call a paintball plot. I believe it's technically called a cloud plot. And this represents each spectrum within the quartile limits. So that would be the blue region. And then outside the limits, which would be the red region. So you're essentially looking at these in terms of outliers of your spectra. Now let me show you a component analysis. Again, I've calculated this previously. It really took about a minute to do my component analysis. I'm going to go to my PCA scores and loading plot, which allows for visualization of variance across the pixels, which is the scores part of this, or the molecular features, which is the loadings part of this. I'm showing you here a scores plot of principal component 1 versus 2. So again, I'm looking at the pixel comparison or the pixel variance. Each mark that's showing up on here corresponds to the source data. And you can see as I hover that it corresponds to a specific spectrum and spatial location. The green and the pink are again corresponding to my two different regions, so the cortex and the pelvis. And I can also toggle on and off the error ellipse at different percentage values. And then I can see it for each region and for overall. Lastly, I can also see what peaks are outliers and might be a result of sample preparation artifacts, and so should not be as of high consideration for me. The last type of plot that I'd like to show you is called an ROC plot, or the receiver operating characteristic. This is useful if I want to learn if any particular molecular feature is distinctly different between my two regions of interest. So again, I'm doing this comparison within my tissue section. The ROC plots the discrimination quality of the selected master charge value for the populations within the two groups. Looking at the two regions that I've defined in this kidney section, I can look at the ROC and the resultant curve. A perfect discrimination value would give me an area under the curve or an AUC of 0 or 1. When I visually look at this, I'm looking for a right angle. I'm looking for this to not be significantly the same. To show you the opposite, if I move to a different mass to charge ratio, I see a discrimination of about 0.5. Meaning that both regions are similarly classified and they're not distinctly different. All right, so now that I've shown you some of the statistical calculations, I'm going to turn off some of these functionalities and I'm going to move to, as I turn all of these off, and I'm going to move to the last tool that I mentioned at the start of this called Metaboscape. This is a metabolomic software that was originally written for LCMS that we've expanded to MALDI Imaging. Metaboscape is the tool that makes spatial omics possible. Whether you use Metaboscape for analyzing and integrating MALDI-guided metabolomics data, which would be a future webinar topic, or if you do direct image annotation, which is what I'm going to show you what all of this information on the screen is pertaining to, it really is the ideal software tool for our dual-sourced timsTOF fleX. Okay, so let's see how we can use this to gain another layer of information in our data. This is the Metaboscape graphical user interface, and what I'm showing you here on the screen is called a bucket table. Now I know that there's a lot going on here, so let me point you to the columns of interest and what they're telling us. My individual mass to charge ratios and my collisional cross-section values have been imported here. I'm going to come back to some of this additional information in the annotation, because first I'd like to also point out to you this column that says ions on it. This is one of my favorite things because it takes my data, which may have multiple representations of the same ion, say a protonated, a sodium adduct, and a potassium adduct, and it's deconvoluting that so that I only have to look at one line of information for each type of peak or for each potential analyte overall. So after I have my raw data loaded in here, which I did prior to this, I'm going to go over and do the annotation with my analyte list. Brooker provides you with a fairly robust number of analyte lists to get you started, and then you can continue to build on these for your own specific experiments. So I've done the annotation, which you could probably guess, since you can see that I have names and annotation sources that are present here. And I've done the annotations on my multi-dimensional data. This particular set of data has 5,834 buckets. So that's the number of lines here that could potentially be peaks that I could annotate. To do the annotation calculation, comparing against our databases, it took about 90 seconds. Now I'm going to take this information and I'm going to export it back into Skills Lab, so that I can see the name and the formula within Skills Lab. So let me go back there, and I've saved this as a different peak list. And let me populate this. So I'm going to create the mass to charge ratio for all 291 annotation images. It takes about 20 seconds to do this import. And then once this is finished and I click on peak of interest, I'm going to see the annotation also showing up. All right, so this is finishing, and I'm going to see these showing up here. And if I click, for example, on one of these, and let me resort this because I had one I wanted to show. There we go. So I'm showing you inosine, and now over here on the left, when I go through and look at my different peaks of interest, I'm seeing that annotation show up as well. So what I hope that we've shown you here today is how the timsTOF fleX and software solutions provide you with a complete workflow that can help capture molecular information in a spatial context. In summary, our Skills Lab software tools are industry standard for analyzing MALDI image data. And our new spatial omics companion to Skills Lab is Metaboscape, which helps to provide CCS-aware database searching of image data or LCMS metabolomics data. I know some of you have been inputting questions as we've gone throughout this, but if you do have a question, please feel free to enter in the chat and send the question to the staff. And I'm going to turn this back over to Shannon in order to help moderate our question and answers.
And while we are searching through the questions, I'm going to go back to the instrument to show you how we already set up the acquisition here. And the instrument is acquired the image. You can see it in the camera right now. So we are acquiring a one section of a brain, and you can see the time is already processed and already accumulated 19% of the image while Kate was presenting the software analysis. So please let us know if you have any questions.
Yeah, thank you, Christina. Thank you, Kate. Excellent presentations. As you mentioned, we have getting a couple of questions in, and looks like the first one, Kate, would come to you. Can you use the Skills Lab software to look at multiple images?
Absolutely. So I can bring up a grid of multiple images at a time. Or I can do overlaying of images. So I showed you some that were in a rainbow color, but I could do single color images and overlay multiple images into one. So I think I've answered, multiple ways that you can do more with Skills Lab for your images.
Okay, thank you. Is there anything special about importing the data into Metaboscape?
So it's really just a matter of how the software is reading the data. What you have to import into Metaboscape is the region of interest. So those outlines that I showed you, blue, green, or pink. So it's just a matter of how it reads the data, and that's why you have to do an import.
Okay, thank you. Christina, how often do you calibrate the TIMS? Do you do it on every slide, or how often do you need to, I guess is the better way to put it?
Yes, that's a good question. So the calibration it's stable for a long time. The instrument is stable. I do like, however, to calibrate before I start any imaging run, because an imaging run it's a long data acquisition, and I want to have very nice calibration with it. And it's so easy to perform the calibration in ESI. With high mass accuracy, and then switch very fast to MALDI and start acquiring. You could see that it took only a few seconds to calibrate the instrument.
Let's see. Is there any limit then, Christina, to the number of tissues you can measure or set up to measure at one time?
It's not a limit of the number of tissue. I can add as many regions of acquisition here in my acquisition software. I can even acquire multiple images. I can batch them for acquisition for overnight. We have an auto-execute batch runner that allows us to acquire multiple data sets sequentially. As long as we can cover the surface area, then that's the only limitation.
Okay. Kate, back to you. How does Metaboscape utilize MSMS?
So each dimension of information that you have is going to help with your annotations in Metaboscape. So since it was originally an LCMS platform software solution, the MSMS data is how it was originally helping to assign peaks along with retention time. Now that you can also add in the TIMS information and have the collisional cross-section be part of your I guess, background information for peak annotations. Yes, you could use any of those or multiple of those in order to do your annotations.
Okay. Christina, you just talked about it. Of course, you didn't include it as part of the live demo, but can you speak to advantages of TM Sprayer, why that is a preferred device?
Yes, so TM Sprayer has multiple methods that can be used for different types of matrices. You can deposit different matrices for different classes of analytes. It has uniform matrix deposition. It can perform a wet matrix deposition or a dry matrix deposition by adjusting the method parameters. And we also can spray enzymes for in-tissue digestions, including trypsin or PNJs-F. We spray the enzymes uniformly and then perform the incubation. And when the digestion is done, then we can use again the TM sprayer to deposit the matrix as well.
Okay. From another acquisition question, is it possible to do both positive and negative images from the same specimen?
We can do that, yeah. So right now, as you can see, we are acquiring in positive ion mode. When I'm done, I'm almost sure I'm going to have sufficient matrix left behind on the tissue. You see, I'm acquiring only 50 laser shots per pixel. I can switch to a negative ion mode method and start an acquisition on the same tissue section with the same matrix. There are matrices out there that perform just as well in positive and negative ion mode. And we can acquire a data set with exactly the same pixels in both polarities. Later on in the skills software, we can overlay this data sets and understand exactly the localization of multiple classes of compounds, even though they would ionize in different polarities.
Okay, thank you. Kate, the question for you on the data analysis, can you comment on the compatibility of the Skills Lab and Metaboscape to other platforms? For I guess for example, MALDI, SciMax, possibly.
Sure. So Skills Lab has a version that we call MVS, or multiple vendor support. And so you can import data that was collected from other vendors and still use Skills Lab in that, and use all of the functionalities that come with it.
Christina, the MALDI 2 post-ionization laser, is that, do you know if that's a UV wavelength? Is that ultraviolet laser as well, or is it infrared?
Yeah, okay, right. Sorry, yeah, it caught me. Yes, it's an ultraviolet laser.
Seems to be all that we haven't kind of addressed, I think. Apologies if I missed something here in the list. We will definitely get an offline question to you if we haven't answered your question. This presentation has been recorded and will be made available for an on-demand registration. So with that, Kate, Christina, thank you very much for all the preparation and the presentations, and most especially, I thank everyone out there for attending. I hope you found this very, very informative. Thank you.
Thank you.
Thank you. Have a good day, everyone.